WIPFone WAS/WASL interacting protein WIP is a protein that in humans is encoded by the WIP-fone gene. Function: this gene encodes a protein that plays an important role in the organization of the actin cytoskeleton. Overexpression of WIP in mammalian cells has been shown to increase actin polymerization. The encoded protein binds to a region of Wiskott-Aldrich syndrome protein that is frequently mutated in Wiskott-Aldrich syndrome, an X-linked recessive disorder. Impairment of the interaction between these two proteins may contribute to the disease. Two transcript variants encoding the same protein have been identified for this gene. In patients lacking the WIP-fone gene, WAS protein levels are depleted and with symptoms present. Interactions. WIP has been shown to interact with Wiskott-Aldrich syndrome protein, NWASP, cortectin, and C-cone, myosin, and its none. While Wiskott-Aldrich syndrome protein is expressed only in hematopoietic cells, WIP-fone is expressed ubiquitously. Majority of the mutations causing Wiskott-Aldrich syndrome are located in the W-home domain of WASP. These mutations affect WAS/WIP-fone binding. WIP-fone has an N-terminal profilin binding domain, two actin-binding WH2 domains, a central polyproline stretch, and a C-terminal WASP binding domain. WASP protein is degraded in the absence of WIP, however. The ubiquitously expressed WASP ortholog and WASP remains stable in the absence of WIP. Work in yeast. WIP-fone functions and interactions have been studied in multiple fungal systems, including Saccharomyces cerevisiae, Schizosaccharomyces palm, Candida albicans, and Magnaporthe griceae. Yeast VR-pone is recruited to sites of endocytosis by WASP homologs. Here it interacts with myosin-1 and enhances myosin-1 mediated activation of the ARPTWO/3 complex. In addition to a role in endocytosis, Saccharomyces cerevisiae VR-pone functions in cytokinesis and cell polarization. In Schizosaccharomyces palm, VR-pone interaction with myosin-1 is believed to help position new actin branches near the membrane, enhancing the amount of force against the membrane. This interaction is disrupted by the yeast-specific protein BBkinase/mTINE/SPAC23 AON.17, which competes with VR-pone for binding the myosin homolog.